Hello, everybody. This is Mr. Andy, and I just wanted to go over some information with you about what viruses are. So, our big questions that we're going to talk about is what is a virus and how does a virus function? So, what a virus is, it's an obligate intracellular parasite. What that means is simply that a virus is something that exists because of something else. And viruses consist of genetic information. It could be either DNA or it could be RNA, and a protein coat called a capsid, and that is it. And like we've mentioned before, the size of the virus is really dependent upon the size of the capsid because capsids are made out of proteins and the genetic material codes for proteins. Sir Peter Medawar, a virologist, once stated that a virus is simply a piece of bad news wrapped in a protein. So here's a couple of the different types of viral structures, specifically the tobacco mosaic virus and adenovirus influenza, and my personal favorite, the T4 bacteriophage. Now, viruses cannot carry out a life cycle outside of a host cell because they're not made of cells. So viruses are not traditionally considered to be alive. They don't have a metabolism, they're not made of cells, they don't have a reproductive capability outside of their host. So because of that reason, they're considered to kind of be on that cusp of life, but not necessarily living. But it's not that it really matters because we know that viruses exist. So viruses were first discovered by this man named Dmitry Ivanovsky. He was trying to study, this is in the 1800s, he was trying to study the transmission of something called tobacco mosaic disease. And so if you look on the right-hand side of the screen, you see that what he did was he extracted some sap from tobacco with a tobacco mosaic disease, filtered it through a porcelain filter that was capable of trapping bacteria because bacteria are orders of magnitude larger than viruses. He rubbed that filtered healthy sap on the healthy tobacco plants and they got the disease. And so he said it had to be something smaller than the cell that was causing the disease. And eventually he was able to isolate and that it was a virus. So since that time, we've discovered viruses for all domains of life. I mean, a lot of viruses are harmless, but some are really quite deadly. Here's a couple of examples. The herpes virus, something like 60 to 95% of the population has herpes simplex 1, which causes the cold sores that you see. Genital herpes is caused by herpes simplex 2, and something like 16 to 18% of the world's population is infected with genital herpes. A couple of these, measles, polio, and smallpox, fortunately, we have a great vaccination effort and we have been able to essentially eradicate these diseases, although because some people believe that vaccines are harmful and might cause autism, measles is making a comeback. In the Middle East, in Pakistan and Afghanistan, polio is making a comeback, and fortunately, smallpox is still essentially eradicated. Here's a couple of the flu. 1918 flu pandemic killed millions of people. 2009, we had an H1N1 or the swine flu pandemic, and it was thought that that might end up being as bad as the 1918 pandemic, but it never reached those proportions. So, if we talk about what a prokaryotic virus is, a prokaryotic virus is called a phage, which comes from their habit of quote-unquote eating lawns of bacteria on petri dishes. And so if we look at the best known estimates, there's somewhere on the order of 10 to the 31 phages on the planet. And you think, oh, 10 to the 31, that's a pretty large number. Yeah, it is a pretty large number. That is more than the number of stars in our universe. So if we lined all of these phages up, we might have a line about 100 million light years long, which is really, really big. A couple videos. This is what a phage infection plaque on a bacterial lawn looks like. You can see that it's eating through the bacteria. Now, phages are really interesting because of their potential use as a therapy, which I will get into here very shortly. Before we get into that, let's go talk about the two types or kinds of phages. One is called a virulent phage, which goes through what we term the lytic cycle. It's a classic viral life cycle. Infection happens, synthesis, assembly, lytic release, and the assembly of the virus is called autocatalytic. The great example of this is my personal favorite again, the T4 phage, or what we term the lunar lander, because it looks like the landing mechanism that our astronauts used when they landed on the moon in the 1960s and 70s. So step 1, the bacteriophage attaches to the host cell, inserts its DNA at step 2, synthesizes the new viral genome and proteins. Assembly is autocatalytic, and then the cell lyses or bursts, releasing more bacteriophages, which can then go infect more bacterial cells. It's a very short life cycle. Contrast it to what's called a temperate phage, or the lysogenic cycle. The lysogenic cycle is much longer, and in fact, the phage DNA injects its DNA, it circulizes, it then ends up, if we look at right here, it ends up incorporating itself into the bacterial chromosome, becoming a prophage. So we continue around this circle, and it reproduces, cell division happens, we get a new population of bacteria infected with the prophage, and so typically what happens is the lysogenic cycle just repeats itself, and you have a very long-term infection. At some point, there will be some factor that determines whether or not the lytic cycle is induced, some environmental factor. And when the lytic cycle is induced, those new phage DNA and proteins are synthesized because they're autocatalytic, and the cell lyses, the phages are released, and the infection cycle continues over. Now, phage therapy, like I mentioned before, through our use of antibiotics, we have created an environment where we have artificially sped up evolution, and a lot of bacteria are now resistant to the antibiotics that we have. So phage therapy is an opportunity to treat the bacterial diseases using these bacterial phages. So there are phages that are specific to bacteria. And so in the last 80 years, this has been used particularly in the Soviet Union and now Russia, but we just really haven't used it here in the United States. It's considered a new avenue of research because of the fact that we are running out of the antibiotics that bacteria are resistant to, or not resistant to. Let's contrast these with eukaryotic viruses. They're more diverse than phages. They can be DNA or RNA. They can be single-stranded or double-stranded, so we have 4 types of eukaryotic viruses. Many of them have an additional coating called a lipid envelope that surrounds the protein coat. So that lipid envelope gives it another unique characteristic. A DNA virus, if you look on the left-hand side of the screen, the DNA virus will inject its viral DNA. It uncoats, replication happens, you get the transcription and manufacturing of the proteins, it self-assembles, it's autocatalytic, and then the viral particles exit the cell. RNA cells are a little bit different. RNA viruses, sorry, not RNA cells, but RNA viruses are a little bit different. Typically that RNA, whether it's single-stranded or double-stranded, that genome is inserted, acts as a template, messenger RNA is produced, you get the glycoproteins, the capsid proteins, whatnot, and then the virus buds away from the cell. These are just a couple of the classes of different types of animal viruses broken down by double-stranded, single-stranded DNA, double-stranded, single-stranded RNA. I recommend, if you want to look through these, that you pause the video and just kind of look through different types of viruses. Now, 2 examples of the animal viruses that I want to cover very quickly are RNA virus influenza. Influenza is easily transmissible in aerosol form, meaning that our nasal secretions in our saliva, if we sneeze, if we cough, and then we touch other surfaces, we very easily transmit that virus. Influenza is probably responsible for more deaths in human history than any other known virus. That 1918 flu pandemic killed like 18 or 20 million people worldwide. It mutates very rapidly, called an antigenic shift. So you can go get a flu shot, but if it's not the flu shot that covers for the virus that is pathogenic in that year, you're still going to get the flu. Now, here's the naming convention. Probably one of the ones that's most known to you would be the H1N1 or the swine flu. The H1N1 simply referring to the ending of the naming convention, the hemagglutinin and the neuraminidase. HIV, probably one of the scariest viruses in the sense that It mutates so rapidly and there's no known cure. HIV is a retrovirus. The genome is RNA, and RNA viruses do not have the proofreading mechanism of a DNA virus, so that's what makes this mutate very rapidly. Now, what does retrovirus mean? If you remember, the central dogma says we go from DNA to RNA to protein. A retrovirus violates the central dogma by going from RNA back to DNA because it codes for something, an enzyme called reverse transcriptase. It makes a DNA copy of the genome from the RNA, which is then spliced into the host cell chromosome. As I said, central dogma violation. This is what that looks like. And here are a couple of pictures of HIV entering and leaving a cell. So acute infection is kind of like the flu. So when somebody gets HIV for the first time, they kind of exhibit flu-like symptoms, and then what ends up happening, as the virus destroys the immune system, the body becomes prone to opportunistic infections, and so HIV ends up causing a disease called AIDS, or acquired immune deficiency syndrome. So the virus can only be spread through bodily fluid exchange, particularly blood and genital secretions. And for HIV, there's various treatments. We have no known cure, no vaccine. They're primarily available in the developed world. So somebody that has HIV will be taking numerous drugs which interfere with the viral replication process. And they're very, very expensive. Plants also get viruses. We're not going to get too much into the plant viruses. They typically manifest as blotchy pigment patterns on the plant, whether it be on the flower or the fruit. Now, what a virus is not, they're not the simplest infection particles. That title goes to these things called viroids, which are disease-causing RNA molecules in plants that don't code for a protein. So here's an example of an RNA sequence of a potato viroid, something that's going to infect a potato. Probably one of the scariest non-cellular agents is what's called a prion. It's a disease-causing protein molecule with no genetic material whatsoever. So it's just a misfolded brain protein, really. And what ends up happening is, in cows, it causes a disease called bovine spongiform encephalopathy, or BSE. We call that mad cow disease. In humans, it's Crutchfield-Jakob disease, or CJD. In sheep, it's called scrapie. And in deer, it's called CWD, or chronic wasting disease. Here is a little microscopic view of the spongiform plaque in the brain tissue of a person with bovine spongiform encephalopathy, or mad cow disease. Currently, we've heard a lot in the news about something called the Zika virus, spread by some mosquitoes. Was a huge outbreak in Brazil, particularly worrisome with the Summer Olympics that happened in 2016 in Latin America. And it's really linked to something called microcephaly, which, if you look at this picture on the right-hand side here, this baby was born with a shrunken head. And so very, very worrisome virus for women who are pregnant or thinking about getting pregnant. So that is going to wrap up our virus video. So make sure that you can explain why viruses are considered non-living or living, depending on particular aspects of their function, the different types of viruses that utilize the information system of cells, compare a bacteriophage or a prokaryotic virus to a eukaryotic virus, be able to compare the lytic and lysogenic cycles. Why do some eukaryotic viruses have RNA genomes? What is a retrovirus? What is reverse transcriptase? Explain the consequences of viral life cycles on human health, and the relationship between HIV and AIDS, and be able to compare viruses to other infectious particles. So that's going to be the end of the video for now. I hope that you enjoyed this.